Hello, so the next part of genetics and health then is the test cross and pedigree diagrams. So here's some little things we need to remember. We need to remember that meiosis takes place to produce gametes, and gametes have 23 chromosomes per cell. We also need to remember this little fact, that when people do produce gametes, only one chromosome from each pair can go into the gamete. And some keywords we talked about were alleles, dominant and recessive. So we said alleles were different forms of the same gene. So for example, we could have different genes for eye colour or different alleles for eye colour. Dominant was the stronger characteristic, so individuals only need one copy of this allele for it to be shown. So the example was if we had somebody who was big B, little b for eye colour, the big B was brown, so they would have brown eyes. Recessive was the weaker characteristic, and for that trait to be shown, the individual must have two copies of that allele. So if we want somebody with blue eyes, they had to have two little b's. Other keywords we had were phenotype, which was the physical appearance of an individual, controlled by their genotype. The genotype is the combination of a person's alleles for a particular gene. Homozygous meant an individual had two alleles, which were the same, and heterozygous means that they've got two alleles which are different. So this part then is about test crosses and pedigree diagrams. So let's get going. So a test cross or back cross is used in this scenario. When an organism can display a trait in its phenotype, but you don't know its genotype. Let me give you an example. In peas, the pods can be yellow or green. Yellow is dominant to green. So yellow is given a big Y and green is a little y. Therefore, if you have a yellow pod, its phenotype, which is its physical appearance, is clearly yellow, but you don't know its genotype. It could be big Y, little y, or big Y, big Y. Either way, both are going to give a yellow phenotype. So here's what you could do. To work out its genotype, you carry out a test cross with a homozygous recessive and observe the offspring. So this means that in this scenario, you need to take a plant which produces yellow pods, which is the unknown, and cross it with a plant which produces green pods, because they can only be little y, little y. Now this is why you do it that way. So you cross the unknown yellow with green, because that way you can be sure of the phenotype of the green. It has to be little y, little y. So if the unknown is big Y, little y, we carry out a Punnett square and a genetic cross, and this is what we'll get. So down here is the unknown, big Y, little y. Up here is the known homozygous recessive. In this scenario, there will be some green offspring. But if the unknown is big Y, and we cross it, that means that all of the offspring end up with a big Y gene for yellow. So that means there will be some offspring with yellow pods. So the difference here is that if the unknown was big Y, little y, there will be some green offspring. Approximately 50% of the offspring will be green. But if the unknown is big Y, all the offspring will have yellow pods. So to do a test cross, what you need to do is cross the unknown, because it can be homozygous or heterozygous. You take it and cross with homozygous recessive, because you can be sure of its genotype, and then you observe or look at the offspring, and whatever the offspring has, that tells you what the parents were. If we look at this question, when two plants are crossed and the phenotypes of the offspring are determined, we find the following ratios: tall 3 to 1 short. However, the tall plants will have two different genotypes. They can either be big T, or big T, little t. Using Punnett squares to explain your answer, how can the genotype of the tall plant be determined? So, what we want to do is cross with homozygous recessive, which would be little t, little t. So it would have the phenotype short. So we're going to do big T cross with little t, or it's going to be this. So what I mean by this is that we don't know if the tall plant is big T or big T little t. We know that if we cross it with a plant which is short, it has to have two little t's. So all we do, or all we're thinking about here, is a theoretical cross. So if we took this unknown and cross it with two little t's, we would either get results that look like this or results that look like this. So if we do this Punnett square, all the offspring are tall. In this scenario, half or 50% are tall and 50% are small. So again, what you have to do is take the unknown, so we don't know if the unknown is big T or big T little t, and you cross it with the short punt, which is two little t's. You cross it and observe the offspring. So in this case, if there are any small plants the unknown must be big T, little t. The big T can't produce short offspring. The next section of this video is looking at these little charts. Now this is a pedigree diagram. Pedigree diagram shows the ways in which a genetic condition is inherited. So the example we're going to look at here is cystic fibrosis. It's caused by a recessive allele, and it causes an excess of sticky mucus to be produced in the lungs and the pancreas. It's really dangerous because it can cause blockages and mean that people that have cystic fibrosis are more prone to infections in both their respiratory system and their digestive system. But to have it, a person must have two recessive alleles. They've inherited a copy of the faulty allele from both parents. So, in our examples here, we're going to use the letter C to show, pardon me, we're going to use a big C to show normal and a little c to show cystic fibrosis. Now, a person who is heterozygous for the gene will be normal, but is called a carrier of the disease. So a carrier would be big C, little c. They are heterozygous. And what this means for them is that they will not have the disease, but they can pass on this little c to their children, which might mean that their children could get this disease, but they themselves will not have cystic fibrosis. So let's have a little look at this. Now, this is a typical pedigree diagram. Usually you've got different colours to show different characteristics and different shapes to show different characteristics as well. So in this case, a square is male, a circle is female, green is normal, and yellow is a female with cystic fibrosis. So what we can say about these people then is person 1 and 2, they are normal males, and number 2 is normal female. What happens is they have children, they've got 4 of them, and this daughter, who is a normal female, has children with this person who is a normal male. They have 3 children altogether, 2 of them are normal, but 1 of them has cystic fibrosis. So in this scenario, these 2 people up here are basically the grandparents. 3 and 4 are the parents of children 5, 6, and 7. And then this relation would be, if these are the two grandparents, that means that this person is the son of 1 and 2. This is the daughter of 1 and 2. And this is another son of 1 and 2. The relationship between this person and these people, this guy would be their uncle. So it basically shows a little family tree, but it also shows how diseases can be inherited within families. Now, with the information that we know that for somebody to have cystic fibrosis, well, in fact, let's write it up here. So a big C was normal, and a little c meant that they had cystic fibrosis. Now, for this female to have cystic fibrosis, she has to be 2 little c's. That means that she had to get a little c from each parent. Now, as well as that, because each parent is normal, they must have had a big C each as well. Okay, so we can say that 7 has cystic fibrosis. So she must be little c. 3 and 4, they have a child with cystic fibrosis. So therefore, they must be big C, little c. Now, the reason why they must be big C, little c is because they're both normal, but they were both able to pass on this little c to their daughter. Now, what does that mean for the others in this? So, what are 5 and 6? Well, 5 and 6 are both normal, so they could be big C, which means that they both got big C's from both parents, or they could be carriers as well. Now, because they're both green, it means that they're both normal. But because their parents were both big C, little c, it's possible that, let's say for example 5 got this big C from his mother or her mother and a little c from the father, so she ends up being this. But equally as possible, she ends up getting big C from her mother and big C from her father, so she ends up being big C as well. So this doesn't tell you certainly about everyone, but it tells you quite a lot of information about the families. Now, as well as that, if we go even further back, it will also tell us that at least one of these grandparents, or both, had to have a little c, because the little c had to be passed along here to their daughter, who then passed it along to this child. So at least one of the grandparents has to have a little c as well. But because they're both normal, that means that the grandparents could be big C, little c, or one of them could be big C. So it is entirely possible that this one was big C, this one was big C, little c, or vice versa. But we know that somewhere one of these grandparents had to have the little c. So let's look at some questions you could get from this. What's the genotype of the child with cystic fibrosis? It has to be 2 little c's. Because it's a recessive disease, you only get this if you end up with 2 copies of the recessive allele. What is the genotype of the child 7's parents? So 3 and 4. Both must be heterozygous. They're both healthy, but both pass on the gene for cystic fibrosis. So they both must be big C, little c. What are the possible genotypes for child 5 and 6? Both are normal, so they could be big C or big C, little c. What is the probability their next child will be a boy with cystic fibrosis? Now, to do this, we need a Punnett square. So we'll set out the parents, mother and father, their phenotype. We're going to put carrier just because we worked out in one of the other questions that they had to have the genotype big C, little c. Gametes that they produce then will be this. We put it in a Punnett square. Two big C's, big C, little c, big C, little c, little c, little c. Now, this means that they have the ratio of 3 normal to 1 CF, or 25% that the next child will have cystic fibrosis. But we're not done yet. It says the next child will be a boy with cystic fibrosis. Now, because there's 50% chance of having a boy or a girl, you do 25% times 0.5, which is the probability, which equals 12.5% chance that the next child is a boy with cystic fibrosis. Or what you do, 25 divided by 2, because there's a 50-50 chance, which again equals 12.5%. Final question then, what are the genotypes of the grandparents of child 7? So one or both of them must have been a carrier, as they are both normal, but their daughter, 3, has inherited a faulty gene which she passed on to her child. So we know that both of them have a big C each because they're both normal, but we're unsure about the second part of it. Because let's say, for example, the normal male, who's the grandfather, was big C, little c. He passes that little c on to his daughter, who then passes that little c on to her daughter. So the grandmother could be big C, but we're not sure. Okay? All we know is that at least one of them has a little c.